So I want to welcome everyone. As I said, my name is Tanya Greaser. I'm the educator with the Family Services Program for the Huntington Society of Canada. I'm really pleased to be able to offer this presentation today, and we'll jump into it. I want to just start quickly by explaining, for those of you who might be new, who have never attended a presentation before, who the Huntington Society of Canada is and the programs that we offer. So we are a nonprofit national organization with the mission statement to improve the quality of life of those affected by Huntington disease. HD was first described by Dr. George Huntington in 1872. Ralph and Ariel Walker founded the Huntington Society of Canada. And as I said, it's a nonprofit. It is governed by a volunteer board of directors. I've been with the Huntington Society now for 3 years, and had previously was the Resource Center Director for Saskatchewan a number of years back, and absolutely love working with this community of people. The National Family Services Program provides direct support for individuals and families in services and education events such as this, short-term counseling, and a number of printed resources, everything from a carer's guide, a physician's guide, and a number of fact sheets, which are really easy to understand handouts that you can access from our brand new website. We are a large program, the Family Services Program, that is. We have a national director. We have myself, the educator. We have a child and youth practitioner. And then we've got 16 resource center directors, all scattered throughout the country. We work with a number of individuals outside of our organization, and are happy to connect with others in a multidisciplinary care team. So from hospital and community social workers to occupational therapists, physical therapists, of course, family doctors, psychiatrists, researchers, neurologists, speech language pathologists, home care aid, genetic counselors, the list goes on and on. There's lots of other people. This is not an exhaustive list. These are probably just the most common folks that we would have direct interaction with on behalf of working to provide the best care for you and your family members, and making sure that you have the resources and support that you need. So the Huntington Society of Canada.ca is the website. You can use the QR code here to scan and subscribe to our email so that you don't miss out on any publications, events, or updates. And also then make sure that you grab any of the printed resources that you need. You can print them from your computer. You can just use them, download them as well. Please also consider, if you are someone who likes social media, go ahead, follow us on Facebook, YouTube, Instagram, and this presentation as well is being recorded and will be available for you on the HSC YouTube page. So final thoughts, just know that Huntington disease is a disease that changes movement, emotions, and thinking over time. But with knowledge from events like this, and planning, support, resources, people can continue to live with dignity and have quality of life. And we're here to support you on your journey. So with that, I would like to introduce Stephanie Bortnick. Stephanie is a research professional working in HD and neurodegeneration research. She manages the HD Biobank, a Canadian biorepository that collects biological samples for researchers worldwide. Stephanie has worked closely with national and international collaborators, including HSC, to strengthen research networks, support sample and data sharing initiatives, and improve engagement between the scientific community and participants. She has a strong passion for facilitating the connection between Canadians and researchers and the impact that it continues to foster. We're very lucky to have Stephanie with us today and part of the HD community and family. And I would like to welcome you, Stephanie, and I'm going to let you take it from here. I'll stop sharing my screen and let you impart your wisdom.
Wonderful. Thank you so much, Tanya. I'm really honored to be here and speaking to you all. Let me just get started on my screen sharing. 1 second. Oops. And presentation. Wonderful.
Looks great.
Awesome. So thank you so much for having me, Tanya, and for the invitation here. I really do love connecting with the HD community, and I'm looking forward to telling you about all the different ways that we connect to the community as the HD Biobank. So as she mentioned, I am the manager of the HD Biobank, and we are a Huntington's disease biobank at the University of British Columbia in the Center for Molecular Medicine and Therapeutics. And I'm talking today about building bridges between families and research. Now, I know your first question is probably, what is a biobank? Well, Biobanking 101 is the collecting of human biological samples for use in medical research. So a sample is collected from an individual either with HD or without HD. The sample comes to me here at the Biobank, and I mentioned we're located in Vancouver, British Columbia, where we process and prepare the samples and store the samples and share them with researchers worldwide studying Huntington's disease. So who are we? Myself included. So we have two directors. We have Dr. Michael Hayden, which many of you have probably heard of. He is the most cited researcher in the world for Huntington's disease, and he's been a part of the research community since the 80s. We also have Dr. Blair Levitt as our co-director, and he's a practicing neurologist at the Center for HD here at the University of British Columbia. He has also been involved in research for many decades and has partnered with the Biobank since the 90s. My name is Stephanie Bortnick, as I was introduced earlier, and I'm the manager of the HD Biobank. I'm responsible for the logistics for everything from patient outreach all the way to sample sharing with researchers and establishing those research networks. I also have Kyla Javier, who works with me. She is our wonderful clinical coordinator who gathers clinical records to connect to the samples, which I'll speak to a bit more later. And we have Haley Finlay Black, who is our in-house research technician who helps with all the lab tasks and sample preparation. It's a great group of people. So I mentioned that we bank and process samples here for researchers. And these are the types of samples that we house here at the Biobank. We collect blood from which we can bank plasma, which is a byproduct of blood, essentially removing the red blood cells. We bank DNA, which is extracted from blood. And we bank cell lines, where we can study how cells grow in the presence or in the absence of the HD mutation and or different chemicals and treatments. We also collect brain and other peripheral tissues, and that is one thing that makes our bank stand out from many other neurodegeneration banks is the collection of peripheral tissues along with brains. Michael, again, right from the 80s, when we learned more about the Huntington gene itself, we learned that it was what we call ubiquitous, which means it's found throughout the body and is involved in many different biological pathways. And it was thought of that, well, other tissues could actually be affected, not just the brain. So other tissues were collected, and a lot of incredible research has been done on things like somatic instability you may have heard of, and looking for biomarkers, which I'll speak more to in a minute. But that is a very unique aspect of our bank. We also collect cerebral spinal fluid, which is a biofluid that gives us an interesting snapshot into what's going on in the brain. We also collect saliva DNA and sperm DNA, and these help us to understand, the sperm DNA helps us to understand intergenerational instability, which I'll speak to more later. Our bank holds over 38,000 samples from over 5,000 individuals. I mentioned that the bank has been in partnership with the community for over 40 years. Our very first sample was actually collected in May of 1984. And it always excites me when I get to give out that sample even today. It's such a legacy of that donor that they're still propelling research 40 years later. And it's just such an honor to be able to partner with these families for such a long time. So why does biobanking matter for Huntington's disease? Well, just as the HSC connects many different people within the HD community, that's what the biobank does as well. So a donor obviously is hoping and working towards, or hoping that researchers are working towards finding treatments for Huntington's disease. That's what we all want. That's what we're all working together to do. But a donor can't do that themselves. They they need to partner with researchers. They need to partner with clinical trial companies and industry partners. And so we are a part of that. Treatment development requires this cooperation. So a donor gives a sample. It's processed at the biobank here. We share it with researchers. They come up with a discovery that's really interesting. Industry partners move that into a clinical trial, and then treatments are found, and it all comes back around to helping the donor themselves. So we are a part of that, that community connection. So scientific knowledge directly from HD biobank samples has already influenced patient care and clinical trials. This is not a new endeavor, and it's been a part of every step of research in the HD community for decades. We were part of validating the gene discovery and part of finding that gene. Predictive testing, Michael Hayden's lab was actually was the first lab in Canada to do predictive testing for all Canadians, as well as some international individuals as well. And that started back in the early 90s. The Hayden Lab and the HD Biobank were also involved in understanding generational expansion and inheritance. And again, that was using sperm samples that were collected in the 90s. And newest understanding that has come out of these samples is understanding disease onset and progression via these loss of interruption modifiers, which I'll speak to a little bit later. They've also directly influenced clinical trials. So these LOI modifiers, which I'll explain later, are really important in identification of clinical trial participants. Current drug development targets were also generated in the Hayden lab with these biobank samples in regards to allele-specific silencing. So you may have heard of drug trials that were reducing all Huntington gene within an individual, and unfortunately, there was quite a few unfortunate side effects when you reduce all versions of Huntington. But research that came out of the Hayden lab showed that you could reduce only the expanded gene allele, and current trials are going on right now using those, and they do seem promising. There's also cellular targets that have been found for drug development. So I had mentioned before this idea of biomarkers, and these are small proteins or molecules that are found in biofluids, so blood, CSF, or other tissues that are not the brain, that can help us understand disease progression and onset. And so, for example, or like just to simplify it, biopsying the brain to see how HD is happening in their brain is difficult or impossible to do. However, taking a blood sample and looking for a marker is an easy way to do that. And that's been done with samples here at the biobank. So I mentioned that biobanking is a network of cooperation. We can't do this ourselves, and researchers can't do it without us. We can't do it without families. And so the biobank really is a way to connect all of us together. So it starts with individuals and families wanting to participate in research. They contact us here at the biobank. We collect clinical records from neurology clinics, genetics counselors, the individuals themselves. We partner with social workers across Canada and worldwide. When people want to participate in research, they know they can contact us. Advocacy organizations like the HSC plays a huge role in helping educate patients about biobanking and about the research that's being done with those samples. And then researchers themselves. Obviously, I need to give these samples to researchers and ensure that they're propelling research into treatments for HD. And we have both Canadian and international partnerships in all of these areas. So what is the HSC's role? Well, we're very, very grateful to be able to partner with them. They've been incredible in helping us with patient outreach, so through webinars and presentations, as well as being invited to the national conferences. I'm always impressed with the HSC's ability to communicate research findings. Anytime I've spoken to the HD research community, and we've done tours, for example, with the BC chapter group, I'm always impressed with the HD community's understanding of the most up-to-date research going on. We are asked the most intelligent questions that really shows that there's such a deep understanding and a deep passion for this research in the community. And the HSC has also been invaluable in generating research funding for groups here at UBC, as well as groups around the world. A, a researcher may have an amazing idea for something to test, but unless they have that funding and are able to do the experiments and request the samples, it's not possible without funding. We had the pleasure of hosting Shelley Redman and Alexander Maxim here at the Center for Molecular Medicine and Therapeutics. We had great tours with Dr. Blair Levitt's lab, Dr. Michael Hayden's lab, and Dr. Mahmoud Pouladi's lab. And we had the whole BC HSC chapter there. We also had fun participating in the local HSC Vancouver walk. There's a picture here of one of our researchers, Inez, with a BC chapter representative, Gail, and it was just such a great time of connection. So what does our donation process look like? I know you're probably thinking, well, you're all the way in Vancouver. How do we do this? Well, that's what I'm here to explain to you, is that we have a donation network for all Canadians, which I'll explain in a minute. So our donation process it starts with education. It starts with learning about biobanking and understanding that we are an available resource. And then it goes to a telephone interview. You reach out to us. We talk about the clinical data we collect clinical data and demographic data, as well as family history. We partner with you in helping you understand how to donate a sample in your local area. And it really is just building a relationship with you and with your family members as we go through sample collection. A sample will be donated. So I will go through the logistics of how to donate a sample in your local area, but we do collect blood, tissue, postmortem tissue, saliva, CSF, and sperm. The samples, as I mentioned before, are then shipped here to the biobank, where they're processed and stored to best ensure their integrity and the longevity of the sample, to ensure they're the highest quality samples for researchers. We also connect digital clinical records to go along with the sample. And again, that's another unique aspect of our bank, is because we've been running for so long, we have incredibly detailed clinical records of individuals and their family members over a long period of time. We then share samples. We create cohorts for researchers based on their research needs and their research questions, and we collaborate with researchers worldwide. I always joke that I'm not a dragon hoarding samples in my freezer. I need to share them. A sample in the freezer is not helpful for research. It needs to get into the hands of great researchers. So we do have a network of national patient sample collection. We've reached most provinces and some of the territories, and we're still actively expanding this network. So if you see your local area doesn't have a lot, please still reach out to me. I am actively working on expanding. So we have blood collection services across Canada, where you go and you get a blood draw, just like you would if you were getting a blood drawn for your doctor. And then we have many wonderful pathology partners across Canada, both in urban and rural hospitals, that will do the postmortem tissue collection for us. And these are just wonderful teams that are really proud to join with us to facilitate these very generous donations. And then saliva and sperm donation is available with an at-home kit to make it very easy for people to participate if they're not currently serviced by the network. However, if you notice that you're not currently serviced by the network, reach out anyways, and I will make it happen. So blood collection, I mentioned before, it's as simple as going and getting a blood draw, as if you were getting a blood draw when your doctor requests one. It's a very professional service. We use the services of phlebotomy clinics like DynaLife, DynaCare, Life Labs. And just to be clear, there's no cost to you at all for donating samples. Whether it be the collection or the shipping or anything, we handle all the logistics of it. You can also participate you can also participate in HD research from the comfort of your home. So the saliva and the sperm collection kits can be mailed directly to your home. We include return mailing supplies and postage. So you provide a sample, you put it in the mailing supplies that we give you, and you drop it off at your local FedEx office. And then we do tissue donation. So this is when a loved one passes. We will collect their brain and or peripheral tissues, depending on how the family feels about it. We handle all the logistics of the donation and any costs associated with the donation. And I just want to say that we do make this process as easy and as smooth as possible for the family members. We understand that it is a very difficult time when you lose a loved one, and there's already logistics with funeral homes and paperwork and such, and we walk you through every step of the process. So it starts with we transport the donor to the local hospital. And when a donor passes, they can pass in hospital, in a home care facility, or at home if they prefer. And we use licensed professionals in the area. So the same transport companies that funeral homes use. So it's a very professional and comfortable service. And we're available 24/7 for these tissue donations. I am called anytime somebody across Canada passes and would like to donate their brain. Then the tissue donation occurs again at a local hospital to you. And we work with an expert team of pathologists at the hospital who will collect the tissues, do initial preservation of them, and then send them over to the bank for further processing and sharing. And then we have the donor transported to your family's funeral home of choice for final preparations. It is one of my greatest honors to walk through this process with families when their loved one is passing. I'm often in contact with the family for, a month or months before somebody passes. And I do try to give as much support, both in the logistics and trying to understand the process, as well as just preparing them for what happens next. And so we've had some people reach out and thank us for what we're doing, and I'm just going to read a couple. So we had a woman who is 66 who passed, and her sister wrote to us, the HD Biobank team were strangers to me before I called asking if it was possible to have my sister's brain tissues donated for research towards a cure for Huntington's disease. But they became a major source of support and comfort to me during this difficult time, and I will forever be thankful to them for that. We had another woman who was about 73, and she was involved and her daughter wrote to us saying, I'm convinced a cure is around the corner because my mom always told me so, and thanks to people like you, it will be. And then we also had a man who passed and donated his brain. He was 67, and his wife said, it was always the donor's wish to donate his body to science, and you fulfilled his dying wish by making it happen on very short notice. Words cannot express how grateful we are to you and how comforted we are to know that the donor is helping with research. And like I mentioned before, I am just absolutely honored that this is part of what I do and part of my partnership with the HD community. So once a sample is collected, as I mentioned, it comes here to the biobank, and there's many different ways that we prepare the samples as researchers need them. So as I mentioned, we do plasma extraction, cell line generation, DNA extraction, which this picture here is one of my favorite coworkers. His name is Eugene, and he is our DNA extraction machine. So we can put in many different sample types and he will extract DNA for us, sorry. He shows up to work every day, just raring to go, and we love him for that. And his name actually is part of the HD community. The HD, sorry, the individuals that were at the HSE conference this last year helped us to name him Eugene, and it was just really fun. From the DNA that's extracted, we do a lot of CAG sizing, and we do a lot of tissue preparation to get the samples ready as researchers need them. A clinical records collection is an important part of biobanking. In-depth clinical records, along with a sample, are an invaluable resource to the research community. So for clinical records, we collect things like demographics. Are they male? Are they female? How old are they? Family history. How many generations has HD affected? Is this a new family? Age of onset information, disease progression, and autopsy reports to understand the pathology going on in the brain. That information, along with multiple samples over time, either from the same donor or from generations of their family members, are just incredibly important for research discovery. I always say, if I hand a researcher a blood sample with no information, what are they going to do with that? They have a blood sample in their hand and they're like, okay. But if I hand them a blood sample and I say, this blood sample is from a 73-year-old male who had onset of Huntington's disease in their 40s. So they've had a very long duration. Their mother passed of Huntington's disease when she was 60. And so then the researcher starts to go, oh, well, that's unusual. It's unusual that he's had such a long progression. Are there other samples that are like that? Are there other people that we could study like that? And it sparks an interest in those samples when researchers have questions about the samples themselves. And so that's why it's really important to collect this clinical data. And again, I mentioned for generations, we've been collecting this clinical data from donors and their families themselves, as well as their clinicians when we have permission from the family to contact their neurologists, their genetic counselors, to really get the most robust clinical information. And then it's really important to get these samples out there, to get them used, to really spark research. So here, UBC has an incredible HD research program, as I mentioned before, with Dr. Hayden, Dr. Levitt, and Dr. Pulati. And they all have many, many research collaborations worldwide that have been going on for decades. So we share samples with those scientists. We often are sharing information about the biobank at many scientific conferences around the world. I joke that we go to every conference of every letter of the alphabet. Also, the HSC has been wonderful in helping us reach the researchers that they're partnered with and through their research network. And then we do a lot of industry outreach, so letting industry partners know that these samples are available, especially with all of Michael Hayden's contacts within industry partners. He is at the forefront of really driving research and academia results into drug treatments. So we have been giving away a lot of samples for research. So in 2025, we were able to distribute over 2,300 samples worldwide in Canada and Europe, in the US, to industry and academic researchers studying HD and other dementias. So what kinds of things are these samples being used in currently? Well, academia has been really great at finding and targeting new mechanisms of the disease. You know, what is going on? What's the biological processes? As we start to understand that, we can find things to target. So We have a researcher that's currently trying to study what cell types are the most affected in the brain early in the disease before symptoms show. There's another researcher that's looking at how our bodies uptake different minerals and how that might be inhibited in HD tissues. I mentioned there's a researcher really interested in our lung tissue, which again is incredibly rare to have lung tissue from HD patients. And they're finding that it may be impaired due to the HD expansion. And they're trying to understand, is that correlated with why HD patients tend to have end-stage pneumonia. And then industry is wonderful at taking that knowledge just steps forward into drug discovery and treatment research. So I'd mentioned before that allele selective gene silencing is being used by multiple clinical trials. And so what that is I mentioned before, it's you have two alleles, one that is what's considered typical, and then your expanded allele. Well, how can we shut off just the expanded allele and leave the typical HD gene to function as it should within our body? They've also been really interested and pursuing this biomarker validation. So trying to understand these small molecules that we can find in our body that may give us an insight or show a glimpse into what's going on in the brain. The HD biobank samples have also been really important in clinical findings and finding information that helps neurologists and genetic counselors better inform and treat their patients in the clinical setting. So again, coming back to those biomarkers, they're all the rage right now in research. And then one that I want to really specify is that there's recently been found a modified gene sequence in the Huntington gene itself that's separate from the expansion that may cause a 13-year earlier onset of the disease. And I'm going to speak to that a bit more right now because it's really at the forefront of research at the moment. So you'll notice these slides look a little bit different. These were generously given to me by Dr. Jessica Dawson, who works here in the Hayden Lab. And this work has been done by Dr. Jessica Dawson and Haley Finlay Black, our research technician. So we have always been told that the CAG repeat length influence disease penetrance, so when it starts and who's symptomatic, and age at onset. So I'm sure you've all seen this table or a table similar that says if you have a CAG repeat of less than 27, you're unaffected. That's typical within the population. If you have between 27 and 35 CAG repeats, that's considered an intermediate allele. You still are unaffected, but there is a risk of expansion in subsequent generations. If you're between 36 and 39, you're in what's called the reduced penetrance category, which is a very, at the moment, a very gray category. So you're told that you may remain asymptomatic, you may never be affected, or you may have onset very late in life. And then if you have a CAG repeat size of greater than 40, that's considered fully penetrant, and you're told that you will have an onset of HD within your average lifespan. We are also all told that the inherited CAG repeat length, so that CAG size, so for example, 41, is the primary determinant of HD age of onset. So you may have seen this, or you may have been told, with your CAG size of, let's say, 44, your predictive age of onset is about 45. Now, let me clarify. It is a very big range of onset. It is not a clear, like as it's shown in this, it's very 44 onset, or sorry, 44 CAG is a 45 onset. No, there's a very gray range that clinicians will definitely let you know about. However, when we are looking at huge cohorts of patients, like we can here at the HD Biobank with our 5,000 individuals, you will find some people are outliers. They have either an earlier age of onset, so they fall below this graph, or a later age of onset, they fall above the graph. And it's really interesting to study these patients to try to understand disease progression and mechanisms of what could be going on. And so that's what we did. In 2019, we were really looking at these earlier onset patients to try to understand what was going on. And in that at that time, there was a change that was found. And so these are genetic changes due to variants in the Huntington's disease gene. So within the gene itself, so whether it's the expanded or the unexpanded allele, you will see in a typical HD gene these interruptions. So you'll notice there is the red bar, which is a CAG, and it has an N value. So for my example, I'm going to say it's an N of 41. And then you have this little CAA interruption, and then you have the polyproline region downstream, so this blue region with another interruption that is a CCA. So I'm going to show these as going forward as beads on a string. So you have your red beads. So this person, I'm going to, as an example, say they have 41 red beads. Then they have this light blue bead, then a red bead. Then we have the dark blue bead. So we've got one individual blue bead, a yellow bead, and then X number of dark blue beads. So you can think of these CAG interruptions almost like speed bumps. You have kind of like two speed bumps there. So you can lose either both speed bumps or one or the other speed bump. There's 3 different versions that we've found with these HD Biobank samples. So in this example, we've lost both speed bumps. We've lost both the light blue bead and we've lost the yellow bead. And what's really important to note is if you notice that blue bead, it's not like it gets cut out, like cut and pasted somewhere else. It actually turns into a red bead. So what's really important to note is that that person that was a 41 red bead is now actually a 42 red bead. And then we can also lose just the light blue bead and not the yellow bead. And with this one, I'm going to show the exact same thing, that this person went from being a 41 red bead to a 43 red bead, which is important, and I'll explain in 1 second. And then we have the CCG LOI, which is losing just that downstream yellow bead and keeping the light blue bead. Now, this person still stays a 41 red bead repeat. So the reason I am highlighting that there's a change in the CAG size is because current diagnostic testing does not look at these detailed sequences or the detailed beads. It only looks at the number above that blue bead. So somebody who has 41, but this either the CAG, CCG LOI or the CAG LOI is told that diagnostically they have 41 repeats. However, when we look more closely, they actually have the 43, and that can change their onset and their progression, just as we know from previous data. However, when we looked closer at this, even taking into account the extra CAG repeats, we actually still found that the people with these LOIs, I'm just going to call them LOIs, all 3 of them have a significantly earlier onset than expected, up to 12 and a half, 13 years earlier, which was quite a surprise for the research community and has very important clinical implications for families. So this identification of the LOI variant is particularly important for those in that reduced penetrance gray range. This could give them a lot more information and understanding about whether they may or may not become symptomatic. So that CAG, CCG LOI, if we look at the entire spectrum of HD patients, so all the way from 36 repeats to 52 repeats, they're only found in about 1% of symptomatic patients. So an important discovery, but also we're thinking, okay, well, they're not affecting that many people within the HD community. However, when you start to look at those in the RP range, in 29% of symptomatic HD patients, so patients in that range that were told they may or may not go on to have the disease, and then they go on to have the disease, 29% of them have just the CAG, CCG, LOI variant, which, okay, so now we're looking at quite a few more people within the HD community. Then if you look at those with a CAG of 36 or 37 that become symptomatic, 75% of those individuals have one of these LOIs, and that is significant. There have also been very a very small population, but there have also shown a few people that have had a 34 or a 35 repeat go on to have the disease because, like with these LOIs, because that 34 is actually a 36 and the 35 is actually a 37. Now, this has important implications, especially in genetic counseling, because, as I said before, the person that was a 34, 35, 36, 37 is now 2 up, still in the reduced penetrance range, but more likely to be symptomatic. And those that are a 38 or a 39 with one of these LOIs are now actually in the fully penetrant range. They will go on to have the disease in their lifetime, and they're not currently being told that because these are not being found diagnostically. So there's lots of implications in this very new and up and coming research. Well, I say new, it has been found in 2019 and extensively studied by many, many research groups worldwide. But it's new to the patient community. What's known in the research community now needs to be shared with individuals and their families. Again, because there's implications. So it may change family planning decisions. It may require early management of motor decline to improve patient care. And the Hayden Lab, especially, as well as other groups worldwide, are really pushing for updated diagnostic sizings to determine these variants and provide correct sizing to patients and their families. So I mentioned that the HD Biobank is a bridge between researchers and patients and their families. This is a great example of that. So in the interim, LOI variant research testing is available for HD patients in the Hayden Lab through the HD Biobank. So we are now working with patients and their families and their clinicians to test for these LOIs in patients where it could be possible or it could affect their decision making. So especially those in that RP range. So to get involved, the clinician or the patients themselves will contact the biobank. We will send an outreach letter and consent forms, and to both the patient and their clinician. We need to make sure that there's clinician involvement. The biobank is a research-based testing site, and so I can't share directly with patients and their families, but we can share it with clinicians who then have the best education and understanding to share it with families. Once documentation is received, patients are sent a saliva kit or a blood draw requisition. And then the kit is returned, we do the LOI testing, and then we send the report to clinicians to help inform the individuals who donated samples. So we're that bridge right now until diagnostic testing standards are updated, which hopefully will happen, but we all know how slow the healthcare system can move sometimes. So another part of the biobank is actually mentoring the next generation of HD researchers, and this is a part of my job that I take great pleasure in, is we often have students, but whether university students or high school students, come into the lab and learn about what we're doing and just spark an interest in that next generation. So the HSC has been wonderful in funding 3 of our summer research students. We've had one in previous years, and we actually have 2 this year that are doing just fantastic cutting edge research over the summer. And I will say one of them is interested in both being a nurse and participating in HD research in a clinical sense. And after working with us, she started as a high school volunteer. And after working with us, she really is now pushing more towards that clinical research nurse to be able to help with clinical trials and things like that. So it is really cool that we can help bring really great people into the HD community through this education. So yeah, we've had another great time was we had a medical trainee come from New Zealand to work with the biobank to understand how patients can be involved in research and how we are integrated with the Center for Huntington's Disease at the University of British Columbia. That was a really important aspect for her that she wanted to bring back to New Zealand, and how to get patients in clinic involved in research programs. So it's been a wonderful time with these students. I want to celebrate with the HD community that there has been recent federal recognition for HD research here in Canada. So we recently received some limited funding from Brain Canada Foundation to help fund the biobank for the next 2 years. And so we shared with them our vision for this Canadian biorepository or Canadian Biobank for neurodegeneration research, and showed how helpful the storage and collection of these records is for the HD community and for neurodegeneration as a whole. And they recognized how important that is, and they are funding us. So we're really honored to be a part of that. So I've given lots of information, and you're thinking, hey, that sounds really interesting. I want to be a part of that. How can I get involved? Well, you can contact us. You can send me an email at hd.biobank@ubc.ca. You can call me. My phone number is 604-875-3811. There's a QR code here that leads you to our website, which has a lot of great information to learn more about biobanking, but really just give us a call and we're happy to chat and help you get involved. And then I just want to say thank you from the bottom of my heart. Thank you to the individuals and families that have put their hope in research, that are putting their hope in research for the future, that have done it since 1984. We are honored to partner with you. Thank you to the researchers, both in history and present in the Hayden Lab. Thanks to Dr. Jessica Dawson and Haley Finlay Black for the information they've provided. Thank you to our institute and funding sponsors, the Center for Molecular Medicine and Therapeutics, Center for Huntington's Disease, the hospitals, and Brain Canada. Now, I think I've left enough time for questions. And yeah, if anybody has any questions, please ask. If you're interested in how to donate in your local area, in tissue donation, in blood donation, I'm open to any questions you may have.